The diseases are the behavioral and physiological changes induced by the pathogens in an organism. All diseases have specific symptoms and characteristics. Similarly, silkworms are also affected by various types of diseases caused by protozoa, fungi, bacteria and viruses, since they cause substantial financial loss to the industry. So, their prevention and control assumes utmost importance. Now, let us see the diseases that affect silkworm. Due to the operation of food chain and food web relationship in natural ecosystem, all organisms have antagonistic relationship with parasites and pests. The effect of parasites and pests is more keenly felt when a commercially exploited organism is subjected to diseases and pests. Due to centuries of domestication of life, silkworm has no natural resistance and also shows neither morphological nor behavioral adaptations to escape parasites and pests. So, man's attempt to evolve a disease resistant variety of silkworm has been only partially successful. No race of silkworm is completely immune to diseases, but Japanese races are believed to be more resistant than others. Generally, healthy and hygienic rearing condition reduces the incidence of diseases. Rearing room and appliances should be disinfected with the 2% formalin, that is 800 ml per 10 meter square area, or 2% bleaching powder prior to rearing. Eggs should be subjected to surface sterilization by 2% formalin. Silkworms are mainly infected by protozoa, bacteria, viruses and fungi. Let us now get to know about each one of them in detail. Protozoan diseases. The protozoan diseases of silkworm are called pebrine because of the characteristic pepper-like black spots appearing on the infected silkworm. Causative organism. This disease is caused by the infection of the protozoan Nosema bombaceus. So, let us know about the mode of infection. The Pasteur recognized three modes of infection, that is oral, contact and transovarial transmission. The life cycle of the parasite is completed within a single host. The silkworm, there are two stages in life cycle, a resistant spore stage, which is the infective stage, and a growing or vegetative stage. The life cycle is completed within a week in temperate regions and in 4 days in tropical region. All stages of mulberry silkworm are affected by the pebrine disease. Some symptoms are pebrine affected eggs. The infected eggs have insufficient or irregularly deposited glue and hence get detached from the egg cord. Overlapping of eggs in the cord indicates pebrine infection. Eggs fail to hatch, then presence of pebrine affected larvae Early instar, the stage between morning, larvae succumb to the diseases and die, but grown up larvae may survive up to pupation. The symptoms of the diseases appear only in the late stage of infection. In the early stage of infection, pebrine can be inferred by loss of appetite, unequal growth, irregular molting in the rearing bed. In the late stage of infection, black spots appear on the body of 4th and 5th instar larvae. Spinning larvae spit and waste the silk without properly spinning. A cocoon in India, the appearance of the typical black pepper like spots is rare in the infected larva. Instead, irregular brown patches appear. These patches indicate the presence of dead hypodermal cells due to pebrine infection. Pebrine infected pupa. Though infected cocoons appear normal, the pupa inside show symptoms of pebrine infection. The body is swollen and black in color. Black spots are present on the sides of the abdomen. Pebrine affected moth. The symptoms are irregular decolorized patches are seen on the body and wings. Scales fall off easily, unstretched and deformed wings, and the control measures are rearing only disease-free laying prepared in grainage after mother moth examination, removing diseased larvae from the rearing tray and burning them, disinfecting rearing room and rearing appliances with 4 to 5% formalin or bleaching powder after pebrine infection to kill any spores that may be present. Bacterial diseases, that is flacheri. Formerly, all diseases with the symptom of diarrhea and vomiting were collectively called flacheri and were believed to be caused by bacterial infection. Later on, it was found that infection of bacteria were not always associated with the flacheri. In view of these points, bacterial diseases, formerly called flacheri, is now categorized into 4 different diseases: bacterial flacheri, septicemia, Scoto, coat. Let us get to know about each one of them in detail. Bacterial flacheri or grassery injury, flacheri disease, considered to be caused by multiplication of bacteria in the alimentary canal, which has become weakened due to bad environmental condition. The causative organism for these diseases include the various pathogens has been suspected to be the causative organism in the initial stages of the disease. Streptococci, coliform bacteria are found, and in the advanced stage approaching death and in dead worms. Proteus group of bacteria have been isolated from the diseased larvae. The red or brown color of the dead larvae is due to the growth of the Proteus bacteria. The infection may be through spread, though oral, and induction by bird rearing conditions. The symptoms of the diseases include the diseased larvae show the following general symptoms of the flacheri and loss of appetite, sluggishness and retardation of growth and softening and inelasticity of the skin. The other symptoms by which the disease can be diagnosed from the other bacterial infectious are shrinkage of the body after molt and refusal of feed, diarrhea and vomiting in the fourth and fifth instar. Excreta with intestinal mucosa, softening of the body and death of the carcass, turning black and emitting foul odor. The control measures are provision of optimum environmental condition and providing good quality leaves during rearing, avoidance of unhealthy larvae by incubating eggs, disinfestation of rearing house and appliances with 2% formalin. Septicemia is the bacterial infection of the hemolymph is the cause for the disease. The causative organisms include Bacillus, Streptococcus and Staphylococcus. Rarely do bacteria infecting the alimentary canal destroy the gut and enter into the hemolymph and cause septicemia. Septicemia is infected through skin wounds. Symptoms of this disease includes softening of the body, change of color to brown, larvae produce excreta in liquid form, prolix lose their clasping power and death of larvae are the symptoms of the disease. Control measures include good sanitation, removal of infected larvae and handling of larvae with care so as to avoid skin wound are the common control measures. Choto disease is caused due to toxic substances entering into the body. The causative organism includes Bacillus thuringiensis or Choto or Bacillus chata. The pathogens secrete a toxin which enters the hemolymph from the gut by absorption. The toxin affects the nervous system and causes paralysis to the larva. Choto disease spreads orally. The symptoms include lack of appetite, sluggishness, shrinkage of skin due to diarrhea, loss of clasping power of legs, death due to paralysis, color change to black after death, internal organ becomes liquefied and digestive canal becomes discontinuous and emit foul odor. Control measures include providing good sanitation, clean leaves for feed, removing dead and infected larvae and cleaning the contaminated rearing room with boiling water, which initiative the toxins or the control measures we can adapt to check the Chato disease. Coat disease is called Ranji in India because of the colors produced in the dead larva. Serratia marcescens, Bacillus protagnosis bacterium causes a fatal infection only when inculcated and in harmless when taken orally. Larvae and pupae are affected by this disease. Infection is through skin wounds. Symptoms are affected larvae are flaccid. After death, the body color changes to crimson red. Control measures include clean hygienic rearing, isolation and destruction of diseased larvae, disinfection with the 2% formalin prior to next rearing. Let us now discuss about the prominent viral diseases. Infectious flagellum was formerly the disease was classified along with the bacterial flagellum. Causative organism being the Morator virus, Kavas 1974 named it as IFV. It differs from the other viral infection in that it does not form polyhedra. Mode of infection being infection is spread orally. Incubation period is 7 to 12 days. Early instars are more susceptible to infection than later ones. Symptoms of this disease include vomiting of digestive juice, body looks transparent and shrinkage of body are the common symptoms of the disease. identification of the virus by histopathological or serological techniques. Gattin disease is also known as the diseases of clear heads. Causative organism for this disease, being a submicroscopic virus, is a primary agent, and Streptococcus bombicina is a secondary invader. The characteristic symptom of the disease appear only by the combination of viral bacterial infection. They are lack of appetite, vomiting of alkaline clear ropey liquid, and accentuated diarrhea. Besides, the anterior part of the alimentary canal is free of mulberry leaves and contains only the digestive fluid, and these areas appear transparent and give the name the clear head to the disease. Control measures for this include maintenance of proper sanitation and disinfection between rearing are the two control measures we can adopt. Grocery disease is called jaundice as the infected larvae appear yellow in color and excrete white feces filled with the viral polyhedra. There are three different kinds of polyhedrosis disease: nuclear polyhedrosis, midgut cytoplasmic polyhedrosis, and midgut nuclear polyhedrosis. These three diseases can be distinguished from each other on the basis of the kind of virus infecting, its shape, size, location in the body, location in the cell, etc. Let us now get to know about various fungal diseases. The fungal diseases of silkworm are called muscadine. The characteristic feature of these diseases is the mummification of the infected larvae till and after death and deposition of calcium oxalate salts. Hence, this disease is also called calcino. Different infesting fungi produce different colored spores, and accordingly there are white muscadine, black muscadine, yellow muscadine, and red muscadine, etc. So, these differ from each other. Also in the tissues they infect, but the general modes of infection, the symptoms as well as the control measures are the same for all muscadine infection. Let us know about the modes of infection. 90% of muscadine infection occurs by penetration through the cuticle, and the rest 10% alone occurs through the mouth or through the spiracles. The common symptoms are loss of appetite, lack in growth resulting in unequals in the rearing bed, oily specks and spots on the skin which may have black or green margin, diarrhea, vomiting, shrinkage of body, and inelasticity of the skin. Dead larvae with white mycelia at first, which later changes to black, green, red, yellow or grey with the formation of conidiospores. Some control measures are: the muscadine attack is noticed, control measures have to be taken both during rearing and in between successive rearing. The infected larvae and litter must be removed immediately before formation of conidiospores, and them and burnt infected and healthy larvae can be separated from each other by separating a net with the fresh leaves over the rearing bed. When healthy ones alone crawl up to the net. Provisions of good ventilation and low moisture in the rearing room. 0.4% to 0.8% formalin is mixed with the burnt paddy husk in the ratio of 1:10 and spread over the rearing tray from 30 minutes for 1 to 2 days, particularly after the molt. Let us go through various insect parasites. The maggots of the tachinid or woozy fly are endoparasitic in the viscera of mulberry silkworm. Silkworm infestation by this fly has been recorded from China, Japan, Thailand, South Korea and India. It was prevalent mostly in northern India, but since 1980 it has been appear in most of the southern states. Nature of damage. If eggs are deposited on the early instar larva, the silkworm dies before pupation within parasitic maggots wriggles out of the larvae. If the eggs are deposited on a late larva, the larva may spin a normal cocoon, but the cocoon is rendered useless for both rearing and reproductive purposes. Crop loss due to woozy fly is severe and as much as 40% reduction in yield is reported. The symptoms are as the surface of the larva shows black spots at the place of entry of the larva. The eggs may be seen attached to the surface. The fly maggots may be seen crawling on chandrige and the rearing room and in cocoon storage places in rearing units. Cocoons may be pierced by maggots. The control measures are: some control measures include to prevent woozy fly from entering the rearing room, a nylon net of fine mesh called woozy fly net is spread all around the rearing room, rearing stand, leaving 2 to 3 feet all around. An additional net may be placed at the entrance of the room as a precaution. To prevent woozy fly from oviposition, the following recommendations are given: spraying of woozicide, a procedure of CSR and TI, Mysore, which kills the eggs ovipositor on the larvae body at the specified doses. Dusting of lavigated china clay through a muslin cloth at the rate of 3 gram per 100 spinning larvae before molting or 4 gram per square feet of the mountage is taken at a protection against woozy fly infection. Chiaso fly or Japanese woozy fly, Sternia sericaria or Crocosmia sericarnia lay the eggs on the lower side of the mulberry leaves along veins. 3000 to 500 eggs are eaten by the larvae. They hatch out when acted upon by the digestive juice, penetrate the gut and enter the nervous ganglia. Then they leave the ganglia, become attached to the spiracle and feed on body tissue. Pest of silkworm. Some pests of silkworm are dermestid beetles, straw mite, ants, lizards, rats, squirrels and birds. Let us know about each one of them in detail. Dermestid beetles. These are the pests of cocoons. The larvae as well as adults of these beetles feed on animal matter including tough leather and silk. They pierce through the cocoon by feeding on them and eat the pupa inside. They are particularly attracted by the smell of the stuffed cocoons and are commonly found in the storage chamber of reeling units. They enter dark and hot environment of cocoon storing areas. The adults are small beetles with brown to black elytra and club-shaped antenna. They lay their eggs on some living matter like stored or stifled cocoons. The larvae are small, fusiform, the pupae are naked. Control measures. Some measures to be taken are cleaning of rearing house and storeroom frequently, Rejected and perished cocoons must not be stored. Cleaning of cocoons, storing trays before and after emergence of moths, repeated turnings of stored cocoons to check pest infestation, the straw itch mite, Pediculitis ventricosus. This mite is an ectoparasite of powder post beetle, rice borer, etc., which is accidentally transported to the rearing house along with bamboo and straw while rethatching and attacks silkworm larvae. The male mite is small and dies day after mating. The female mite attaches it to the silkworm larvae and pupae, draws nourishment from the body and also produces a toxin which kills the host. Symptoms. Some symptoms of this disease are silkworm larvae affected by mite lose appetite and become inactive, experiences difficulty in excreting. The excreta are attached to the form of a bead, vomits green fluid and excretes black fluid. Pulsation of the dorsal vessel becomes lower and irregular. It dies. Control measures are: building material must be carefully inspected, wheat, rice and straw should not be stored nearby, and application of acaricides to the rearing room is recommended. Ants. Ants attack silkworm in the trays. For prevention, use of ant well below the rearing stand is useful. When the ants attack is at the time of spinning, a little kerosene can be poured on the floor around the legs of the chandrike or bamboo mountages, two of which are usually kept slanting in the form of inverted. Hexera mermis and microamphidis attack silkworm, particularly the young ones. It penetrates into the body and kills them off. The affected larva becomes transparent at the head and silky white in the posterior end. Lizards, rats and squirrels. These are some of the larger vertebrates which pick up larvae and cocoon and may be prevented by putting wire mesh on all the windows and providing ledges around the rearing house. Birds such as crows and sparrows pick up the silkworm and the mountages are kept out of doors at the time of spinning. Damage by these predators can be avoided by keeping the mountages indoor at the time of spinning. Conclusion. In this session, we have discussed various silkworm diseases, insect parasites. We have also discussed about pests of silkworm. To summarize, development and extension of sericulture has been accorded high priority by the government for alleviating unemployment and rural poverty. It has been recognized as an important rural industry. Silkworm is susceptible for a number of diseases and pests, and these significantly reduce the yield of cocoons and cause great loss to the rearers. So, this pocket gives an account of protozoans, fungal, bacterial and viral diseases, insect and non-insect pests, parasites and predators of silkworms and measures for controlling them. Since the mulberry silkworm is a domesticated variety of silkworm, it is susceptible to diseases and attacked by pests and diseases, parasites. Silkworm rearing is continuous in tropical areas and mostly seasonal in subtropical and temperate zones, lasting from spring to early autumn. And since silkworms are reared year after year, This may possibly help the perpetuation of the pest and parasites of silkworms. Resistance or susceptibility to a disease and pest is only comparative, and there is no silkworm race totally resistant or immune to disease or pest. Many of these diseases and pests not only affect the physiological process, but very life and existence of the silkworm itself, causing great trouble and loss to the silkworm rearers. The importance of silkworm disease and pests in order to susceptibility successfully raise the commercial cocoon crops cannot be overemphasized.